Kubermatcher here. In the late 1990s, marine biologist Stephen Haddock paid a visit to fellow scientist Oshimu Shimomura at his laboratory in Woods Hole, Massachusetts. The two researchers shared an obsession with bioluminescence, light produced by chemical reactions in the bodies of living things, most famously the firefly, but also in fungi and multitude of ocean creatures. At one point during their meeting, Haddock recalls, Shimomura poured what appeared to be large sesame seeds out of a jar and into his hand, dribbled some water onto them and crushed them into paste in his fist. Then he shut off the lights. His palm glowed a transfixing blue, as though it held a fairy. The sesame seeds were in fact the dried bodies of tiny crustaceans known as ostracods. In 1954, ocean bioluminescence saved one military man's life. At the time, future Apollo 13 astronaut James Lovell was a fighter pilot. He was on a training mission off the coast of Japan in stormy weather, when the instrument panel in his cockpit suddenly short-circuited. All the lights and dials went dark. He could no longer rely on technology to point him back to the aircraft carrier. Looking into the blackness, he noticed a shimmering green streak in the sea, which he realized was the ship's bioluminescence wake. He used that trail of living light as a lodestar and managed to land safely. The following year, Shimomura began his own affair with bioluminescence, one that would eventually revolutionize the field of biology. In 1955, Shimomura joined a lab where he was tasked with extracting the organic compound luciferin from ostracods and determining its precise molecular structure. Today, scientists know that in many bioluminescent organisms, an enzyme named luciferase catalyzes a chemical reaction between luciferin and oxygen, which produces light. But back then, we didn't fully understand how it happened, Shimomura says. It was a mystery. After 10 months of labor in the lab, he became the first person to crystallize luciferin, an essential step in studying its structure. In the 1960s, he continued his research at Princeton University, where he started to investigate the luminous jellyfish Aquaria victoria. Shimomura and his colleagues collected numerous specimens and strained them like cider apples to obtain a small amount of pure glowing squeezate. Within the shining liquid, they discovered a protein named aquarin, which releases blue light when it reacts with calcium, even in the absence of oxygen. Another protein in the jellyfish, green fluorescence protein, or GFP, sometimes absorbs the blue light and releases green light in response. In 1978, after collecting nearly 1 million jellyfish, he had thoroughly elucidated the structure of aquarin and the nature of the unique light-generating reactions. Both aquarin and GFP, as well as a gene code for the latter, became indispensable tools in biology and medicine. Scientists could now tag and observe the intricate dances of previously invisible genes and proteins in living cells. In 2008, Shimomura received a Nobel Prize in Chemistry for his work on GFP. So, bioluminescence is a beautiful thing. Kubermatcher here. Ciao.